Robert Field, and it's yours.
Okay, let's see where we are here. Okay, can everybody see that okay?
Yeah, we can see that.
Yeah, okay, super. Okay, so I'd like to start by thanking the organisers for the opportunity to tell you a little bit about some of the algal work that we've been doing over the last few years now. Work that started originally in Norwich and some of which is now in Manchester. So the work is predominantly activities kicked off by Ellis O'Neill and Sue Kuhar-Dunlop during their PhDs. They both now hold faculty positions, as does Ravi Singh, who is a postdoc in my lab and who's gone back to India to a faculty position as well, aided and abetted by a variety of collaborators in Norwich and beyond. I realise I'm in the translation session, so rather than talking about glycobiology in general, carbohydrate biology, I'm going to focus in around carbohydrate active enzymes. Why did we want them? How did we go about it? And where is it taking us in relation to Euglena? So we very much started conscious that while Euglena produces a variety of metabolites that are potentially of interest for a variety of reasons, whether that's as vitamins or as potential therapeutic leads and the like, or indeed as materials or energy sources. We were focusing very much in around paramylon itself, a storage polymer of glucose. So our reason for getting into this in the first place is that I'd spent the best part of 10 years working on starches in plants, getting very, very frustrated by cereal genetics and how long it takes for wheat to grow. And we went looking for something simpler. We stumbled into Euglena thinking that this would be relatively straightforward, and that was a big mistake. Nonetheless, there are many and varied opportunities that have come up from this work, and I'll just share a few with you. So as one does these days, the first thing one wants really is sequence information about genome or transcriptome for a variety of reasons where others in this audience are better equipped to judge than me. We've gone down the transcriptome route, looking at both light and dark grain Euglena, and we were able to identify just over 20-odd thousand genes expressed in the light, 20-odd thousand in the dark, constituting about 32,000 overall. So it's almost as though Euglena has two chassis with a completely different set of metabolism going on, depending on whether this is light or dark grown. Of the 32,000 genes, only 14,000 or so match anything that's in the database on a BLAST search. So there's an awful lot in here that we know nothing about, was the conclusion. So from a carbohydrate active enzyme perspective, clearly we're expecting a wide variety of different enzymes. And indeed the top hits from the annotatable genes from the transcriptomes that we generated came up with hits from all of the major kingdoms of life. So we really were expecting diversity all round. Looking specifically at the carbohydrate active enzymes, there are the major classes of enzymes are the transferases, which basically stitch sugars onto things, and the hydrolases, which take them off again. So we were quite surprised to see just how much commitment Euglena makes to producing proteins in these families, as well as a variety of binding proteins and esterases as well. So if we look at this in practical terms, just plotting the number of hydrolases against the number of transferases, the plants feature in the top right corner here. They make a lot of carbohydrates, funnily enough. Below the line we have Ascomycetes and Basidiomycetes, wood-rotting fungi and the like. Again, not surprising that they're predominant hydrolases. The orange circle here is Euglena. Its phylogenetic neighbors, loose phylogenetic neighbors, the Leishmania and trypanosomes, are down here. And the one closest to Euglena is in fact us, humans. So we were a bit disturbed to find this single-celled organism having, in principle, similar capacity for glycobiology as humans. That was not what we were expecting to see. So just to give you a very brief snapshot of what some of these enzymes look like, I'm just going to pick out odd examples. So what I've done here is to plot glycosyl hydrolases here, the number of genes in a given family above the line for Euglena and below the line for humans. And just to pick out some predominant ones, GH3, 5, and 81, which are found in Euglena but not in humans. These are all predicted to be associated with beta-glucan metabolism, either exo or endoglucosidases. The question really is, if you've simply got one beta-glucan, your paramylon, why do you need quite so many copies, 18 copies of this gene? So we're not quite sure what the answer to that is at the moment. Looking at the transferases again, just to pick out a couple, GH23s, these are fucosyltransferases transferases that modify end-linked glycans typically and have an impact on the ability of those glycans to be released from proteins. GH77s are typically plant cell wall biosynthetic enzymes associated with xylose and arabinose metabolism. So we have some information about genes and what they might do, but in practice, what glycans are present in Euglena. Here, with help from William Willett's lab, what we've done is two things. One is to do a sequential extraction of carbohydrate, array the materials, and then probe with either monoclonal antibodies or lectins of one description or another. As you can see, the predominant signals here in the middle are all associated with binding of a beta-1,3 glucan. The odd thing, sorry, is that we seem to be picking it up in flagella, which we didn't expect. LM6 here is the arabinan, which maybe ties in with our previous notion of the GT77. We've also looked at what's on the surface of the organisms, and what we can see is that the only lectin that dramatically cross-links this organism is right at the bottom left here. RCA is a beta-galactose binding lectin. So there must be beta-galactose on the surface of this organism, that's for sure. With ConA, a mannose binding lectin, we sometimes see some staining when we use fluorescent ConA. We only tend to see it at the sites of cell division. Now, there's a lot more work needed before any meaningful biology comes out of that, but it is known that in mammalian cell division, you get clustering of glycolipids around the axis of cell division. So maybe there's something there to be followed up on yet. In terms of the structure of the Nglycans in this organism, again, what are they? Well, they are actually remarkably simple. They're very predominantly N-linked high mannose glycans. So not complex like higher animals, for instance, but much simpler than that. They do contain some additional functionality, the amino ethyl phosphonates here, which you find in things like T. cruzi. In relation to paramylon, again, we have a plethora of potential enzymes that could contribute to the generation and degradation of the granule. We found it difficult where we looked at candidate genes to express them heterologously, particularly in E. coli. I'll come back to that. One enzyme that frustrated us enormously was the beta-1,3 glucan phosphorylase, which was documented 30 odd years ago, but we could not find a candidate gene in the transcriptome. In the end, after a lot of playing around, we actually went back to a bacterial enzyme from the family GH94. We had to use a second algal strain to make any progress. So what we've in fact done is to take the GH94 bacterial queries against Ochromonas transcript to identify hits, use those hits with the Euglena transcriptome to identify hits in Euglena. In practice, what we've demonstrated is, in fact, these are three different classes of carbohydrate active enzymes, all operating on similar substrates. The sequence identities are low or not detectable. So it's not surprising that blasting with bacterial sequences, we didn't pull anything out of Euglena. These are all very similar at the overall architecture level, even though the sequence level is very weak. The issue with expression of the Euglena protein we took up by a metatranscriptomic approach by taking candidates out of a metagenome based on thermophilic microorganisms, products being much easier to express. And bizarrely, although these enzymes classes have very little similarity at the sequence level, at the structural level, at first glance, they look rather similar. The difference of two inserts here, one here and one here, on the enzymes that work on the oligosaccharides. And what this does is basically open up the active site. It removes some of the gatekeeper residues and stops them regulating the size of the substrate. So although there's no sequence identity here, we can still see at the structural level that there's a lot of conservations, which adds complexity all round. I won't go through the fine detail there.
Excuse me, Rob, you have 1 minute left.
Yes, indeed. So these enzymes operate on beta-glucans, but can we get them to do anything else? Well, yes, indeed we can. Rather than feeding glucose 1-phosphate, the bacterial orthologs here that will, of the Euglena proteins, will transfer galactose, glucosamine, and mannose onto glucose-based acceptors. Why is that of interest? Well, that allows us to access molecules that look like the human milk oligosaccharides. Now, these have a variety of beneficial effects, whether it's prebiotic stimulating commensal microorganisms, blocking adhesion of pathogens, or indeed impacting on the gut immune system and on inflammatory responses more broadly. So infants that are breastfed, for instance, are much less susceptible to Campylobacter infection than are those fed on formula. So there's a lot of interest on how we can access these enzymes. Euglena is providing us with a suite of enzymes that can help in this manner. So it's not just about the weird and wonderful biology of Euglena, it's a huge resource of enzymes, and more particularly genes to go searching for things that are easier to express if you're a novice at that kind of thing like us. I haven't spoken about it today. There is a huge capacity for natural products biosynthesis as well. And perhaps Paul Zimmer might say a little bit more about some of those molecules. With that, I will close and thank you for your attention.
Thank you, Rob. Unfortunately, because of lack of time, we have to move on, but there is already one question for you in the chat box. So please answer it there. And our next speaker is